Okay, clear to all of you? Now moving further, I'll show you one more type of diagram, a sketch of this antibody. See here, this is more clear to you. This is the constant region of the heavy chain. This is the constant region of the light chain. They are bound by disulfide bonds here, and this is the variable region of the light chain and variable region of the heavy chain. Variable region of the light chain and variable region of the heavy chain. Clear to all of you? Just take a look on this diagram for 2-3 seconds, then we'll move further. Special mention to the antigen binding sites which are present here at the tips. The variable region of heavy chain and variable region of light chain provide antigen binding site and are specific in nature, unique, different, new for each unique, different, new antigen. Clear? Okay. Now the next topic is types of antibody. We have 5 types of antibody. See the diagrams here. IgG, we call it as immunoglobulin. I will tell you about immunoglobulin also in the subsequent chapter portion. So you can see the variable regions here. You can see the diagrams. There is difference in the colors. That means all these are having different amino acids. So IgG, IgE, IgD. Ig stands for immunoglobulin. So immunoglobulin, sorry for that. Immunoglobulin G, immunoglobulin E, immunoglobulin D, immunoglobulin A, and immunoglobulin M. So IgD, IgD, IgG, IgE, IgM, and IgA. Okay, so they may be simple as these 3. These are the simple ones and these are the complex one, IgA and IgM. Why they are complex? Because here many IgE or different kind are bound together by disulfide bond and joining of the chains. Here 2 different types are bound together by bonds, by some kind of disulfide bonds also, and chain is being joined. So you have to know about all these types of antibody, their specific function, their process of mechanism of attacking is not in your syllabus. So you just have to understand that there are 5 types and remember the name. Understand Ig stands for immunoglobulin. I'll be telling you about immunoglobulin in the next part of the chapter later. Now the next topic of discussion will be the classification of antibodies on the basis of their mode of action. That earlier which you have studied was on the basis of their structure. Now here you will understand how they are being classified on the basis of their activity. The first is antitoxin. Antitoxin, as the name says, antitoxins are produced in response to the toxin and neutralize the toxin. So this is one mode of action. How they are acting? They are producing antitoxins are produced in the response of any toxin which is being produced by any intruder and neutralize them. That means antitoxins are produced in response to the toxin only. Okay, they are not going and affecting the bacteria but the products of the bacteria. Second is agglutinins. Agglutinins clump a particular antigen of the bacterial suspension. If there are so many bacteria in the suspension, they are going and attaching to a particular antigen and clumping with it. They clump, they coagulate it. Okay, so their mode of action is coagulation or clumping. The next is precipitin. They precipitate and form large complex with soluble antigen. These precipitin kind of antibodies, they go and combine with the soluble antigen and form large complexes, and these large complexes precipitate and stops the activity of the antigen, kills the antigen. Okay, next is lysine. Lysine means lysis, that means breaking down. They disintegrate or break the bacterial cells only. They go and disintegrate the original thing, that is the bacterial cell only. They go and they are not acting on the products which the cells are producing, on the products of the bacterial cell, but lysine are going and disintegrating the whole bacterial cell, so they are not able to produce any toxin substances. The last one is opsonin. Opsonin combine with the surface components of the bacteria and phagocytosis occurs. They engulf the whole bacteria inside. So these are the 5 mode of action. Just a quick revision again. Antitoxin produced in response to the toxin which are being produced by the bacteria. So bacteria produce toxin, and in response to the toxin our body produces antibodies which are functioning like antitoxins, and they will neutralize the toxin. Second is agglutinin. They clump, they coagulate. Their major mode of action is coagulation. They coagulate the antigen of the bacterial suspension. Okay, not killing the bacteria but coagulating the antigen which is produced by the bacteria. Next is precipitin. They are going and attaching to the antigen itself, forming large complexes, and because of being large in size they precipitate and they subdue the activity, they reduce the activity, they stop the activity of these antigens. Next is lysine. Lysine, they disintegrate or break the bacterial cell only. So when the bacterial cell is there, lysine is going and acting on that particular bacterial cell and kills the whole bacterial cell. So origin is only lost. Origin is only being killed. Clear? The last one is opsonin. Opsonin combines with the surface components. They go and join the surface component and slowly and steadily engulf the whole bacteria inside it and digest it by the process of phagocytosis. So these 5 fundamental things, 5 fundamental mode of action of antibodies you should remember, because these all things, whatever I have told you, are all coming in examination, are all important as far as examination is concerned. So I think I have told you enough for today. We will stop here, and just I have shared the notes over the GCR. Just see the notes. If possible, try to copy as much as you can write down, so that once you write down the things it becomes more clear to you. And yes, it is compulsory for all of you to draw the diagrams. I can accept the photostat copies being pasted in your notebooks, but as far as diagrams are concerned, you all need to draw and practice the diagram and submit it to me before the next class. Okay, so we stop here. See you in the next class. Thank you so much.